Hi everyone. Whoa, that is loud. I'm going to step back a little bit. So welcome to our session. If anybody wants to move forward, we're going to try and make the last half of this as interactive as possible. So you will be hearing our intros, our discussions of our problems, and some solutions we've come up with, and then we will invite you guys to let it all out. Share your frustrations that you've had to deal with, solutions that have worked for you, things that you just now thought up on the fly and think maybe that'll work. We have this notepad that we will be taking notes on, and we will be happy to post it around and share it. But before we get to a main session, I would like to introduce Christian, who is going to tell us about the Finance Committee.
Hi everyone, I'm Chris Lytle from Dartmouth Cancer Center. Nope. Do we have a finance slide up? Doesn't look like it. Okay, we'll go without it. I'm the chair of the Finance Committee here with ABRF. We, of course, are looking for members, looking for people who like finance. There's a good group. I am stepping down as chair after this meeting, and Rich Cole is going to be taking over the chair for that. We're looking at changing how we do investments with outside groups. We're also looking at watching the budgets and proposing budgets, and if that's your type of thing, we'd love to have you join the finance group and contact any of us if you can. There would have been a nice QR code for you to do that, but it does pop up periodically as the slides are going through things. You'll see a bunch of guys, some of my friends who I actually kind of hung out there. We're smoking cigars and drinking scotch, but it was a fishing party that we did right before the Nerds. And I will say none of us would have known each other had we not joined ABRF. So keep up the friendships and jump on some committees and research groups. Thank you.
Thank you, Christian. So before we get into the main presentations, I'm going to introduce myself and the motivation for this session. So I'm Jenn Page. I am one of the co-chairs of the Genome Editing Research Group. This talk is not about genome editing today, though. We're joined with Moli, Rena, and Guru, all also members of the research group, as is Madam President Kim Delventhal, and a couple other people who right now are not in attendance. So the motivation for this session, originally a year ago, Moli and I were talking about how when we come to ABRF, there are a lot of fantastic talks and resources for people who run instrumentation-based codes. So for someone who will never have to write an S10, most likely, I know a lot about the process, and that's great, but we don't hear a lot of things that affect personnel-based or service-based codes and the things that we have to deal with. We are also ourselves customers of other codes. Sorry if I'm jumping the gun here. That will be discussed. But then, while we were trying to come up with topics and things we wanted to discuss, I got a really infuriating email from a customer of mine. And it was right before we were supposed to have a GOG meeting. And rather than be able to go to that meeting and join the group, I said, here's a topic we will need to discuss. It didn't work. So the slide's not up. It didn't work, so I'm not paying. What's really infuriating about that was the project was working. For some reason, the PI didn't know that. We'll get into the other failures for how that was possible. But anyway, we just wanted to share some of our unique frustrations and We are happy to invite you guys to come up again, share your frustrations, things that affect you running personnel calls, and like I said earlier, any solutions that you have that work for you, may work in the future. Hey, what if we all just try this? That kind of stuff. And with that, oh, I also was going to say, while we are all genome editing research group, and I said this talk is not about genome editing, we will be using those as examples for some things. So if that's kind of a disconnect, you can just mentally replace service you provide with genome editing. And I'm sure since biology loves to mess with us, it'll still work. All right, thank you.
Hello, I'm Moli Joshi, more popularly just known as Moli. And I'm going to set the stage for our struggles, and the hope is that we start a discussion, because I've been coming to ABRF for several years, and I have heard a lot of talks about instrument-based cores and how they manage their issues, but I have yet to actually hear a talk which deals with cores like ours, which are more people-oriented. So that is the general goal. Let's get the conversation going. Okay, my disclosure is I do not work for any company, and the opinions that I'm expressing today, they are strictly mine and not affiliated to any institute or even GERG. These are my frustrations and mine alone. You might share my views or not. That's totally up to you. Okay, so I run the functional genomics facility at the University of Colorado's Anschutz Medical Campus. I joined the core as a core manager and a consulting scientist in 2013. Last year, I was promoted to the director position of the core. My core offers several technologies, mostly to modulate or manipulate gene expression and study gene function. So we support shRNA or CRISPR technologies. Since I joined the core, I have actually turned the core from a repository and distribution core into a bespoke turnkey solution provider. So we do a lot of genetic screens, we do a lot of cell engineering, we do cloning, we teach, we do R&D, development, and whatnot. We have been talking about, so Jenn mentioned that we don't use instruments, but that's not true. We basically work with the basic molecular biology research infrastructure. We do have some instruments, but we can still do our job without the instrumentation. So I've been doing this a long time. My core serves 5 different campuses and has supported more than 370 labs and supported like over 270 publications and several grants, clinical trials. And so I think that tells us that we know what we're doing. As I said, we've come a long ways from what the core used to be when it started. It couldn't have been done without my amazing staff. This is a long list of people who worked with me over the past 13 years. But at any given time, we've never been more than a team of 3 people. Okay? And recently, I also took over the protein and antibody production core. So in addition to all the functional genomic stuff that I do, I also now manage a cell line generation core, cell line repository. We do monoclonal antibody production, protein and recombinant antibody production. So now I'm also pretending to be a protein engineer. Okay, so, well, I don't have a, okay. So the new functional genomics is essentially modulating gene function at the DNA, RNA, protein level, and we generate cell line models. Currently, I do this with my wonderful manager, An, and my AAV production specialist, Joshi. But for the Cancer Center, we also partner with the organoid core, and hence we are also known as the Functional Genomics and Tissue Modeling Shared Resource. All of this information, too much information, is just to tell you that everything that we do is very labor-oriented, very skill-oriented, does not utilize large equipment. So I've tried to set the stage, and now this is where my opinions start. Since I've been coming to ABRF, I have realized that there are two types of core. You can argue otherwise, which is fine. So I divide the cores into instrument-based cores, which can be sequencing, mass spec, flow, animal imaging, NMR, electron microscopy, any of those cores that use a big piece of equipment, an expensive piece of equipment. And then the other group is people-based core, which is viral vector core, genome editing core, protein and antibody production, iPSC, organoid, transgenic, animal husbandry, histology, which mostly utilizes people to do bench work, wet lab stuff. We can also include bioinformaticians and scientific writers. They are in their separate group of their own, but I can explain why I think they have their own separate group. Most of my experience is based on this group that is highlighted in green, because that is the kind of core I run, that is the kind of core that I interact with, and that is my experience. So what I'm talking about is based on my experience. So I know every core uses instrument and has people, so we can't really differentiate them in some ways. But the way I differentiate is, in the instrument core, if you remove the instrument, there is no core. And I don't know if people will agree with me, but that is how I am distinguishing these. In the people-based core, there are no instruments. Yes, we can now buy some instruments for automation, but even without the instrumentation, we can execute those processes maybe a little slower. So the instrument cores are typically formed of data generating core. They are really expensive instruments. Not every lab can buy them. And so if you buy and set up a core, PIs have no other option but to come to you to get their data. Generally, one campus has one of the core. Like, I have not come across a campus that has multiple mass spec core or multiple electron microscopy core. So, I've heard this phrase over and over again this year at the meeting: you build it and they will come. So that is the instrument cores. Also, the instrument cores have a lot of options for, well, not lot, but they have options for writing grants to get these instruments. And of course, their main fiscal responsibility is to cover the instrument service contracts, which are also insanely expensive. In contrast, people-oriented cores, there are no big high-ticket instruments typically. They generally work with generic lab infrastructure. Basic research labs with basic molecular and biochemistry understanding can execute these projects, which means that cores like mine have significant on-campus, off-campus competition. And I will get to that. Also, we need highly skilled staffed, highly experienced staff. On a single day, I am culturing maybe 10 to 15 type of different cell lines. I need to know how to work with established cell line, primary cell line, adherent cell line, suspension cell line, neuronal cell line, breast cancer cell line, you name it. I have to edit genes that are regulating gene functions, and so I need to have a lot of experience to execute my services effectively. And the main thing is, there are no grants that I have been able to find that will fund hiring of service professionals into cores like mine. So basically, we fund these people through chargebacks, revenues. Sometimes we can request the PIs to put us, the core, or the staff on grants. Some PIs who understand the importance of our core, they do. but not everyone. There is R50. I did hear somebody mention that they do write a lot of R50 for their core. But from experience, I can tell you, getting an R50 is not easy. I am one of the R50 award recipients. And if you are trying it, come talk to me, and I'll tell you my struggles in getting to a successful point. I'm going to leave the bioinformatics and the scientific writing groups alone at this point, but I'm happy to chat about that. Okay, so the other thing that I do want to mention, cores like mine, we create stuff. I am not trying to step on anybody's toes or say that the other cores are not doing what we are doing, but we create stuff. We physically create stuff. We create novel reagents. We create novel molecular biology tools, disease models that have not existed in the past. And we do that very efficiently, and we do it very efficiently. So at this point, we are in this amazing technological phase where the technology is evolving very rapidly. Technologically, everything is possible. You come to me and say, hey, I want to knock out this gene in this cell line. I would say, yes, it is possible. But biology may not always agree. And that is where sometimes projects don't work, or you don't yield the desirable results. Hence, that is what brings us to this session. It didn't work, so I am not paying. Okay, so we all have had unique challenges related to the services that we provide. The most important challenge that I feel is our services are very time-consuming and very labor-intensive. It's not like putting a sample on an instrument and maybe it will take 24 hours to run. Creating a cell line, editing a gene, creating a cell line can take several weeks to several months. In this time, you have started the work. The PI comes and says, well, I'm not interested, so I'm not pursuing this. I'm not going to pay. So this has happened with most of us. Biology, I've already mentioned, there are some certain things that you cannot modulate. If you're trying to knock out an essential gene, good luck. Biology is not going to support your quest. We deal with difficult cell lines. If you guys have worked with stem cells, iPSCs, primary cells, you probably understand that they are not easy to culture. They are not easy to edit. iPSCs randomly tend to differentiate, or they might lose chromosomes and things like that. So we are constantly struggling with biology while trying to get to our deliverables. There are other issues, and we can skip over those, but happy to chat later. Some other issues are standardization. Every core in every university that does gene editing does it slightly differently. It doesn't mean that any process is wrong. It's just there is no metrics for standardization. But when you compare to an instrument, the instruments come with protocols that have been optimized. You process your sample this way, and you put it on the instrument. There is a good chance that the Illumina platform on our campus and the Illumina campus on Salk Institute, they generate similar data as long as the samples are processed the same way. But Jenn's process of making cell lines or editing stem cells is different than my process because my infrastructure is different than hers. And then, of course, we deal with challenging cell line. There is high lab-to-lab variability. But one of the things that we deal with a lot is the end user skill, knowledge, and experience. Oftentimes, we deliver stuff that is, to the best of our knowledge, what they asked for. But sometimes the PIs and the research, the grad student, undergrads, they don't even know how to use those reagents. And they come back to us saying, you gave us the wrong stuff. Talking about money, because everything is about money, in my case, I'm not an institutionally supported core. we have to generate 100% of our operation cost. I am part of the cancer center, so a percent of my salary and my manager's salary is paid by the cancer center, but it is a very small drop in the bucket. So just to give you an example, we are a very small core. We are 3 people and have always been. There is a lot of math here, but essentially, to keep this operations going, I need to generate at least $21,000 to $25,000 a month, which for a lot of cores, it might be like, this is not a very, big issue. And then when we recruit, it's a problem. As I mentioned, staff skill is something that we really need to retain, because if our skilled staff leaves, it's not easy to replace it. If your instrument breaks down, maybe you can replace it with a better instrument. Yes, financial constraint, but we cannot always find a person with the same amazing skill set to replace. It takes years to get to that point. Some of the strategies that we have come up with to overcome this is based on our experience, we've come up with very detailed intake forms, smart sheets, disclaimers. We let them know that, we are going to edit the gene. We will provide the data at the DNA level. We are not responsible if you still see protein or RNA. There is more biology. So we outline the scope of work in great detail so that we are both in agreement that this is what is going to happen. Errors and troubleshooting. As I said, sometimes it's just biology. If you cannot knock down something, you cannot knock it down. It is not possible. Maybe a few years down the lane, maybe there's a new technology that can make it possible, but we have to deal with biology. But sometimes, there are errors made. We also have staff. Somebody makes mistake, take responsibility. So we are very cognizant of that, and I do take responsibility. And then again, sometimes it's the user. I can give you lots of examples, but not going into that detail. Ultimately, it is about money. Since our projects take so long, if you wait till the end of the project to build, you're going to be out of money for a very long time. So somebody told me, well, try the Lego billing system. So this is my term, Lego billing. So you bill in blocks. For every specific service, you have a price. So when people come to you and say, how much is this going to cost? You build your Lego block and say, this is how much it is going to cost. And as you start executing your project, this is done, you're built. This is done, you're built. This is done, you're built. And so this gives you and the PI a way out. PIs, today they come and say, I want to knock out pick 3CA. And then 2 months later, when you are in the middle of starting single-cell cloning, they're like, you know what, I'm not interested in this project, let's stop it. So you are not completely out of the money because you've already billed them for the work that has been done. Some of the other things, somebody mentioned at one of the talk yesterday to include SR and shared resources, core facilities, and staff in research grant. I have had pushback on that, and I'm happy to discuss that later. Institutional supports are really the way to go because they can help with a lot of things. We are in an ever-changing technological space, so we need to keep up with technology. But if we are always working towards recovering cost, we don't have time for R&D. If we make a mistake, we don't have time for troubleshooting. How do we recuperate money from a failed experiment? Who's going to pay that? Like if I have to repeat an experiment, the PI is not going to pay me a second time to do the same thing. So, if there is a level of institutional support, that would be great. And honestly, I feel that core facilities should be treated more like facilities and administration, like all the NIH dollar money that comes in. We've been talking about indirect cost. I think 1% of that should come to the core facilities, because without the core facilities, these amazing institutions, they're not really going to be able to push science forward. And I would like to get your opinion on that. I'm actually going to stop and let my next speaker talk, but happy to talk about authorships and patents later.
Do you want to do that?
No, I think I went through everything, so you can go next.
You want to do your acknowledgements?
Yeah, I've already thanked my team, but I do want to thank ABRF for giving me this opportunity. There are a lot of people and money. I've tried to apply for every possible grant that I could potentially apply for, and I have been successful in getting those grants too. But that wouldn't be possible for my amazing colleagues at GURG and support from ABRF. So happy to take questions later. Thanks.
Hi, good morning. My name is Rena Lapidus, and I'm the director of a translational laboratory shared resource, and I'm going to tell you exactly what we do. I am the newest member of this gene editing group, and I am the least experienced. I'm going to give you one of those examples, but I'm going to show you that we are definitely a people-oriented, service-oriented core. Let's see, my disclosure is I'm a founder of a small biotechnology company. That's not doing well, but that's life. Okay, so my core, I'm part of the Cancer Center. Most of what we do is cancer. We have 4 goals. We model cancer in vitro and in vivo. We support early phase clinical trials. We offer laboratory support, so if there's a clinician out there who wants to do research, they can come to our space or send their fellow or send their resident, and we can help them conduct their experiments. And we just recently, 3-4 years ago, added CRISPR. We do the easy CRISPR. We just do SNPs and knockouts, because I am not as good as these guys. So basically, in vitro, we have a cell line bank. We do drug testing, Western blots, qPCR, a lot of transductions, CRISPR, knockouts, and SNPs. We do model cancer in vivo. That's probably most of what we do. We do a lot of drug development studies, maximum tolerated dose. We model cancer in vivo, novel drug testing, PK, pharmacodynamic studies, and then we support early phase clinical trials. And what I'm really excited about is that we do have examples where we've done the in vitro work, the in vivo work, and then for that novel drug, we've supported the phase 1. And for me, as a cancer biologist, that's what I call myself, that's my goal, right? That's what I'm hoping to do. So here are a couple of our success stories. This is one example where we created we knocked out a gene, since this is a genome editing group. We knocked out a gene, interferon gamma, in 2 cell lines, a Lewis lung, murine lung cancer line, and a control line for a B16 murine melanoma. And then the investigator herself, actually really cool story. She's a PhD student who was an intern in my lab like 5 years before. And that was another really neat thing about being in academia, is that I was able to actually influence her to go get a PhD in immunology, and now she did this work, and now she's actually working for a startup company. But she actually put it into mice, and she found that knocking out interferon gamma actually changed the tumor microenvironment and the role of the Treg, the T regulatory cells. And she published it in a pretty good journal, a high impact factor. The next example is work that we did. This is 2 different publications. We not only knocked out the gene of interest for the PI, Dr. Montaner, but we also transduced the cell line, and then we injected it orthotopically, which means into the site of the head and neck, into the mouth of the mouse. And we monitored it. After a set period of time, the knockout and the parental, we euthanized the mouse. We imaged over time. You could see in the E that you can see where the cancer is. We then euthanize the animal, take the lungs out, and saw that with the knockout you got decreased cancer. That's another really example of how we're translational, right? We help people from in vitro to in vivo prove their concepts. Okay, so those are my 2 success stories to tell you, but what about when things go wrong? So this is my first example. This literally happened this past week. We do a lot of transductions, and this one just didn't work. And the goal of our transductions mostly, we do most of them are so they can express luciferase or fluorescent proteins, so we can see the cells in their assays or in their animals. We are the core that uses all the other cores, so we had the viral vector core created the lentivirus for us. We used the cell line from our bank. We did the transductions. We sorted the cells in the flow core for green, and then the cells didn't grow. This happened in 3 cell lines in the past week. Now, since I've been here at ABRF, all these wonderful people have been giving me great suggestions about what may have gone wrong and where I need to go find the problem. We did look for mycoplasma. One of the cell lines were tested for mycoplasma. The other ones, we had so few cells, and we had to grow them in such a small amount that I couldn't test it for mycoplasma. But the cells didn't survive. So from Moli's point, right, the flow core got their money, right, because I paid them up front to sort the cells. The viral vector core got their money because I paid them to create the viral vector. And I do bundle all those costs into what we charge the investigator, but we're not going to get our dollars because we can't provide the cell line to the investigator. How many of you have ever had, not that problem, but a similar problem where it didn't work and then you just couldn't get the funds back, right? I see some hands. So how do we charge them? Right? And this is an investigator who uses my lab a lot. So the important is I have to maintain the relationship. I don't want to get him angry. The next one is a CRISPR example. This is, once again, we do mostly cancer, so we're trying to knock out what I think is a large gene, 200,000 kilodalton protein in a triple-negative breast cancer cell line called 231s. So we design the guides, we generate the bulk population, we do the single cell cloning, we do the Sanger sequencing. We think we have 100% knockout. We give the cells to the investigator, they do a Western, they still see the protein. We go back, we do it again, design different guides, right? We still cannot knock out that whole protein. It's really complex, it has multiple start sites, alternative splicing. Once again, being here these last couple of days, I've been getting some great advice about what to do. How do we charge them? You know, she's worried about publishing it. Interestingly, she did do a functional assay, the PI. It works beautifully. She sees the effects she expects to see. But, Rena, we can't publish this because by Western blot, we can still see the protein. So, I've been a core director now for 16 years. I came from industry before that. But our motto is that if it's our mistake, we repeat it for free. And if it's their mistake, then they still have to pay us. Do you guys agree with that? We think that's a good motto, right? That's mostly what we do. I think one thing is challenging is deciding, okay, it really is the PI's fault. You know, and Moli was saying that sometimes we have to rely on their expertise to get it right. I have another example where I gave a knockout to an investigator, and she came back 3 months later and said, it's gone. I said, well, what about, early freeze back? Oh, we didn't freeze it back, and they've passaged it 30 times. I was like, and I thought we did the education. I thought we educated. So here are some of my solutions and some of what we talked about in the last couple of days also is, the first thing we do is meet with the investigator and define the scope of work, right? We do this at person, mostly in person, and we do this at no charge. So we figure out what the scope of work is, we then write it up, and we get an approved project plan. And some people do it more formally. I know Jenn does it more formally, and a scope of work. And then I create a quote, and I give it to the investigator and say, this is how much it's going to cost. What I've just decided to do literally in the last 3 months is to give them a range, because if it's easy, it's one price. But if I have to do it 2, 3, 4 times, sometimes I do, then I have to factor in that range, and I promise that I will communicate with them if it looks like the price is going up. I'm not just going to surprise them at the end that it's a problem. Do you guys have this as well? People catch me in the hallway and they say, oh, do this. Can you do this for me? And I'm like, sure, I'll be glad to do that for you. And then I don't write it down, or I don't send them a quote because, hey, I'm just going to do what I always do. And I'm changing my ways. Every time I'm going to get a quote and I'm going to get affirmation from the PI that this is what we're doing and this is what they're going to charge. This is what I'm going to charge them. And then, so I think my point is communicate with the investigators, all right? I want to talk about disclaimers. My next slide is about disclaimers. I just say no surprises with those investigators. So here are some disclaimers. You guys, I'm not going to read them, but I think this was another one of my errors, and I've been doing this for 16 years, and it amazes me that I'm still figuring out better ways to run my core, right? And you can always use ChatGPT or one of the AIs to help you come up with some of this wording, right? But on all of our quotes, which I now have to be, quote unquote, religious about providing to my investigators, right? I need to have disclaimers like, hey, this quote's only good for 6 months, because what if or when at every fiscal year, I need to reevaluate my rates, and my rates go up. Well, if the project lasts 2 years, because or a year, some of them do, the price has to go up, right? So I think disclaimers are something that I've been missing, and that I think it's really useful for all of us to include on our quote. So projects in progress. Jenn, I stole this from Jenn. She calls it Transparency Tuesdays. So apparently every Tuesday, her core communicates with her users to say, hey, here's the status of your project. We do the same exact thing. We share electronic notebooks, we send data, we give updates. We do a lot of in vivo work. A mouse died, a mouse is losing weight, all this sort of stuff. We communicate because at the end, I don't want someone to say, well, why didn't you tell me that? Well, I'm not going to pay for that because it wasn't what I expected. So we share the results with investigators. Troubleshooting, I include the investigator. It's their expertise. Do you know your protein? Do you know your biology? All that sort of thing. And then I try, as Moli said, about billing with milestones. And that's part of the quote process, too. I need to make sure I get paid as we go. So I've now also started to structure our quotes so that we can get billed stepwise, so we don't lose all the money if the project doesn't work. So the end is we write a project report. The project report could just be PowerPoint. It doesn't have to be a fancy Word document. I did find this website that's project report templates that was a lot of PowerPoint, but I want to give all of the data to the investigators. And that includes stuff like sequences, because how many of you have had investigators come back after 2 years, 3 years, 5 years, or longer and say, hey, I'm ready to publish that now. What were the methods? And then you have to go back and say, oh my God, what was that, right? So I try and give them all the information, and then when they come to me, I can say, I gave you that information, or at least I have it in one spot. Transfer the material and then also transfer the material with the appropriate education. We do a lot of cell culture education, freeze back early, test for mycoplasma. The edits aren't edits, they're not permanent, they're all this sort of stuff. If you have an electronic lab notebook, remains open, and then of course invoice them at the end so you can get paid for your work. And that's the people who do all the work in my lab. I wanted to make sure I give them credit. And I'm moving it on to Guru, and then we can talk. Hopefully all of you can come up and tell us your experiences after Guru finishes speaking.
Okay, my slides are, okay, it's up there. How many of you buy this? I'm sure everyone buys this, right? Every day. And how many of you buy? Why is it not going forward? Okay. How many of you buy bread? I'm sure everybody. How many of you have returned any of these at any time in your life? There is one. Okay. Let's talk about servicing, like roofing service. If you are hiring some people and then if they do a good job after 2-3 months or something like that, you are happy. And sometimes it cannot go well as you want. So what I'm talking about and what we are all talking about, this kind of troubles that we face in our core facilities. And core facilities like ours, particularly that involves heavily skilled people doing very lengthy experiments, sometimes going for like years. For especially generating transgenic mouse models, sometimes it takes about a year, sometimes it can go for 2 or 3 years. So if things don't work at the end, we will get blamed. So let's talk about it did not work, so I'm not paying. So in my core, that is transgenic core, what we do is that we will see what we can do to help. That is the attitude we start with every individual cases where there are conflicts. So let's talk about who I am first, and my name is C.B. Gurumurthy. I'm from University of Mississippi Medical Center. Recently, I moved from Nebraska to Mississippi, that is UNMC to UMMC. There is just a point mutation in my institution name. So I also go by CRISPR Guru on LinkedIn. Okay, so and also I'm very proud member of ABRF for nearly 14 years, and I really love coming to this conference and then meeting people, friends, and then learning how to run core facilities better. So I moved to Mississippi just about 9 months ago, and I launched a core called Genome Editing Research Education and Animal Genome Targeting Core, which stands for GREAT Core. And so right now, it's only 2 people, myself and Martin. I call my core as 2 men and a mouse. And I'm also very fortunate to have moved to Mississippi because the core, my department chair really, really likes core facilities, and he strongly supports core facilities. So let's talk about research and services part of my core. And I want to give this flavor so that like at the end, we can have like about 20 minutes or maybe about 30 minutes to maybe 20 minutes to talk about. So my core facility also does, we also do like research. So there are 2 technologies that we already published on the left-hand side, that is EasyCRISPR and GONAD. And right now, I'm also developing some new technologies, that is like recreating and script, which I'm not going to talk about. And EasyCRISPR and GONAD, I'll just finish them, give you background about it in just two minutes. So using EasyCRISPR and GONAD, we can create large knock-ins up to like 2 kilobases, conditional knockouts, reporters, and all those things very easily. Whereas for multi-kilobase knock-ins, we don't have technologies that are robust enough to be adopted in the core facilities. So what are the types of genome editing in mice? Like there are 4 kinds. One is knockout, small changes, large cassette insertions, and gene replacements. And for the large cassette insertion, for example, is like Cree or reporter lines, and gene replacement is conditional knockout, one of the examples. So for the first 2 category, many people, when the CRISPR was kind of like published in 2012, many transgenic core facilities adapted very quickly to create knockout and small changes, but large cassette insertions and gene replacements was quite difficult. So and also large scale genome engineering, that is like 10, 20, 30, 40 KB, it is quite hard. So we solved this problem, that is like a large cassette insertion and gene replacements using 2 technologies. One is called EasyCRISPR and GONAD. And for the large scale genome engineering, so it is quite hard. We are developing new technologies. So EasyCRISPR, which stands for efficient additions with single-stranded DNA inserts CRISPR. The idea is that like when you want to create knock-in, many investigators or many transgenic core facilities used to use like double-stranded plasmid DNA, and that is homologous recombination. That was not working very well. We used long single-stranded DNA, which changed the equation, changed the balance towards homologous recombination. And we were able to create large knock-ins very easily. And this is the first proof of principle experiment. 3 pups were born. All 3 pups were perfectly inserted with conditional knockout. And so these are like 6 genes. You can see that like a number of zygotes that we injected less than 100, and we were able to generate like some projects with 40% efficiency, some 100%, some 70%, and so on. So most other CRISPR methods required about 200 to 400 zygotes, whereas EasyCRISPR required only about 100 zygotes. So just to summarize EasyCRISPR, if you want to create a knock-in mice with a GFP at the N-terminus, all you need to do is that find a guide RNA that is cutting at the close to ATG, and take GFP and then add about 50 bases nucleotides, homology arm on the left and right, and it's long single-stranded DNA, and you can create large knock-ins very easily. And if you want to do the same thing at the 3' end, all you need to do is that change the donor DNA with the homology arms and the guide RNA. And if you want to create a conditional knockout, top is the wild type allele and the bottom is the conditional knockout allele having 2 LOXP sites. You take 2 LOXP sites, stitch them together in a long single-stranded DNA, make 2 cuts in the genome, drop the cassette in, cassette out, and insert your new cassette. That's as easy as that. And right now, we published this paper almost like 9 years ago, and it is downloaded more than 60,000 times, and it has been cited several hundred times. And we also published this in Nature Protocols about 7 years ago. And that is also downloaded quite heavily, and it is adapted in many transgenic core facilities around the world. So a second technology that we developed is called GONAD. Can EasyCRISPR be done in situ? So like this is the traditional way of making transgenic mice. Like you take about 10 to 15 males and 10 to 15 females, mate them together, collect their zygotes, and then inject one by one. And at the end of the day, you transfer them into pseudopregnant females and generate pups. And so instead of doing all these 3 cumbersome steps, we thought, why not do all the steps in the same mouse by electroporating the CRISPR-Cas solutions into the oviduct within the same mother. And we gave this, this is a proof of principle experiments showing here, and we gave a name called GONAD, which stands for Genome Editing via Oviductal Nucleic Acids Delivery System, which is a microinjection independent genome editing method. So these are the two collaborators with whom I have been working on for nearly 10 years, and we have published more than 30 papers together. Most of them are related to technology development in mouse genome engineering. So this is the GONAD workflow. I'm not going to talk much about it, and let's talk about the service part. So in our core, we offer end-to-end services. What that means is that like, the central part is the main thing that is done, that is a microinjection of zygotes and embryonic stem cell targeting. But on the left side, we added upstream services that say we design animal models. We also do construct building. And at the downstream, we also do genotyping and custom breeding of the mice models that we generate for animal investigators. That means investigators don't have to do anything, and there is a less variability from investigator to investigator, and that way we can have the full control about the project that we do for every investigator. So this is approximately cost about $8,000 if you count about like how many projects we could do with the 2 people per year. And if I assume maybe about 20 projects, maybe about like $200,000 if we have to spend. So for the model designing, for each project, it costs about $3,000 to $4,000, and microinjection cost about $8,000, and downstream cost about $3,000. If you add all of that, most models generation takes anywhere between $10,000 to $15,000. And also it takes anywhere between 3 months to 1 year, sometimes up to 2 years, depending on the complexity of projects. So that means like it is a high-priced item, and also it takes long time, and many things can go wrong in any of these steps. So what that brings us to discussing what are the common types of animal models, what it takes to run a transgenic core, and factors that can help a transgenic core to be efficient and successful. I'm going to finish all these things in about 5 minutes, and then I'll leave about 20 minutes for discussing. So what are the common types of animal models? We already discussed knockout, point mutation, knock-in, insertion of large cassettes, and gene replacements. And commonly used method that we use is easy CRISPR. And what it takes to run a transgenic core, there are 3 important things. One is animal facility, second one is equipment, and third one is skilled scientific staff. So for the animal facility, so you need to have dedicated rooms. Like if you are working in a large common area, so sometimes like those things will have a lot of negative effect in your embryo production, microinjection, and also like a pathogen-free animals you have to create. So then the second thing is like IACUC protocol is very necessary, and you need to have a good relationship with the institutional IACUC committee as well as the animal facility folks. And second one is equipment. So coming to our core facility, equipment is very, very minimal. You just buy basic microinjection setup, and once you buy, they are good for 10, 20, 30, 40 years. And what is most important is skilled laborers, skilled scientific staff. So let me talk about equipment. There are 3 things. One is microinjection, tissue culture facility, embryonic stem cell work that is very rarely done these days, and molecular biology, which is just a basic PCR, gel electrophoresis, and gel documentation system. And coming to microinjection, so you just need this kind of microscope with a setup, and this hardly cost maybe about $80,000 to $100,000 by the this is just one-time investment. And coming to skilled scientific staff, so microinjection is an extremely sensitive and very skill-involved experiment. If you think about like a mouse is almost like just a thumb size, right? Like, and you have to know where exactly the oviduct is there. You have to go to the oviduct, surgically open it, and blow the zygotes out. And then after that, you do the microinjection. You have to implant back into the same kind of oviduct in a second kind of mice. Like, think about it. It is a microsurgery, and that kind of surgery is extremely hard to master, and it generally takes anywhere between 3, 5, 7 years to really learn the good technique on this. So skill sets involved is like embryology, microinjection, and molecular biology, and factors that can help transgenic core to be efficient and successful are skilled scientific staff, keeping up to date with the technology and adapting to change. Adapting to change means like sometimes like technology becomes outdated and then you want to rapidly adapt to the new technology. For example, like when the CRISPR came, like many people were not really able to adapt very quickly, and some of those core facilities closed that I know of about 12 years ago. So, and another key success for Transgenic core facility is like offering end-to-end services. Sometimes like if you rely on investigator-deposited constructs and guide RNAs and things like that, then they may not be very well, they may not be knowledgeable about how to mix those and then how careful they should be in providing us the endotoxin-free and also like these reagents are injected into the zygotes. They are very, very sensitive. So we take all of that into control by doing ourselves. So skilled scientific staff, how many technicians you need? Like embryology, you need 1 to 2 people. Microinjection, you need 1 to 2 people. And molecular biology, you need 1 person. So offering end-to-end services is one of the things that we talked about. And with that, I bring back to the original slide. Transgenic core, we will see what we can do to help. And now let's discuss in the panel.
Okay, thank you, everybody. We'd like to open the floor to all of your comments now. If anybody has any unique frustrations or anything that we didn't touch on, or anything that you would like to suggest, we can share plenty of other quotes that we've heard from PIs about their dissatisfaction with our services, such as the great one, "We want to know who to blame for this." Good attitude, thanks. I don't want to work with them anymore. I don't, anyway. But anybody else have anything that they would like to share? Oh, just want a hug? That's okay, I don't hug either. So, that's all. Oh, yay. We have people approaching the microphones. And also, we can write things down, and it'll make it on the video.
Well, thank you. Very good session. My name is Abhinav Jenn. I direct epigenomics core at MD Anderson. So my one suggestion is to include us in your people table of the people's-based cores. We have these concerns all the time. So I think I like Moli's idea about the Lego-based method. What we do, I don't know if it helps or not, but we have our protocols are, of course, long, and there are a lot of, we created a lot of go, no-go steps in the protocol. So my technicians and me, we review the data at each step. If something does not look right, we immediately create a report and send it to the investigator that what we believe is these are no-go. We can't process these samples anymore. So, because initially we learned hard way. We kept doing it, and it was the same thing. Okay, it didn't work out, so why am I paying for all this stuff? The other thing that we actually also kind of doing is the communication. I'm a scientist myself. I'm very transparent. These are my colleagues. We don't want to spend their money unnecessarily. So we do very clear, transparent communication with them at every step. So I think the modular costing method works quite a bit, and it's not a big hit in their pocket as well, and making it very clear that it may or may not work if you are trying this assay. We also do our own controls with every experiment, because most of these methods need new buffers, buffer replacement and stuff. So sometimes you don't rely the sample. They could question, I've received question, maybe your buffers were bad or something. So we started including controls in every experiment that we did for them, along with samples created in our own lab. So that satisfies them. So I started having less of this question now that maybe something was wrong on your end, because now it's very clear that, so yes, it costs a little bit extra, but we started charging them for control samples as well. So that actually created or deleted this bad feeling out of the way that, you know, it didn't work, so I'm not paying. So they're just paying for what worked. So hope this helps. So thank you.
Thank you. That's great.
Yeah, so great discussion. I've been trying to bring this topic up recently, actually for a couple of years, in terms of bioinformatics. So I know Molly touched on this at a very high level, but I think it's a critical component of this discussion as well. I think bioinformatics, the cost recovery model is very hard as well, because even though we can be in grants, grants are subject to gaps in funding, right? Last year is a good example of that. Grants are also subject to gaps in funding through other ways. So what if a PI leaves a university and goes elsewhere, and you have your bioinformaticians in their grants has other personnel? Because many times they don't want to add everybody from the bioinformatics core as a key personnel. So that can also create a gap in funding where you're suddenly now losing that funding and you have to quickly recover that. And then there are also other challenges that are more bioinformatics specific. That may be something that is linked to cutting edge methods, right? So a lot of times we're tasked with having to analyze the latest of omics technologies, which moves very rapidly, but it takes time to evaluate the methods and really establish the best practices. So how do you, for instance, charge for that appropriately? Do you charge the first PI that huge, right, amount of effort, and then everybody else comes in cheaper because you learned from that first experience? I mean, I've seen people do that, but that's not really the best, approach. I think we can all agree that it creates a little bit of an imbalance. And then there's also scope, right? We, for instance, also write scopes of works, but the final deliverables in any bioinformatics projects are very hard to define it. You may think that you know in advance, but really when you deliver it, many times there's a lot of iterations afterwards, and some groups want you to take the step further to interpret that data, and that's going to be very project defined. So if you're in a grant for 10% effort, but then you're suddenly having to make the biological conclusions, all the figures, so on and so forth, that's no longer 10% effort, that's much more, but there is some resistance in really valuing that. And then a lot of these institutions are not going to want to support that because they think, hey, the PI should be doing so on and so forth, even though it really is an institutional value to at least bring those methods and establish best practices. So I definitely think that bioinformatics does fall under that people category, and it has some unique challenges in there as well. Thanks.
By the way, somebody else should probably write this.